territory of the Lenni Lenape people. This series is organized by the Research Coordination Network for Evolution and Changing Seas Temporal Genomics Working Group. And this is a bi-weekly seminar that's aimed at showcasing research into temporal genomics. By temporal genomics, we mean studies that use genetic samples collected over time. These are often historic and contemporary genetic samples to examine questions relevant to the ecology and evolution of species over the past several hundred years or so, especially their responses to human-induced changes in their environment. We're also really interested in highlighting best practices and new methods for those of us who are interested in doing temporal genomics work too. So yeah, we have a diverse range of topics. We're getting towards the kind of last half of the seminar here. We've got 3 more left. So thank you everyone for coming. We have some people who've been really good in terms of consistently attending. So thanks everyone for coming. We have our seminar today with Nina, and then we have 2 more roundtable sessions where we have a couple of researchers who are a little bit kind of earlier in their careers talking, giving a short kind of summary of some of their temporal genomics work. So be sure to check those out later in June and on July 1st. So also note that we don't have enough time in the seminar series to highlight all of the amazing work that people are doing in temporal genomics. So we have a Twitter conference coming up, which is really exciting. I'm going to put a little link with some info on that in the chat right now. And yeah, we're, anyone who's, especially, people who are in attendance here, if you have work that you'd like to present at that, even, research ideas, feel free to submit an abstract. The abstract submission deadline has been extended a little bit to the 14th of July. So the conference itself is going to be running from July 7th to 9th. So that link is in the chat. Okay, just a few ground rules. While our speaker is presenting, we ask you to please be respectful, keep yourself muted, hold all questions until after the presentation is over. And once Nina's done, we ask you to either use the raise hand button or indicate in the chat that you have a question to ask. Katrina Catalano will be helping me to moderate, so either she or I will call on you, and then you can ask your question. Also note, we do have a lot of registrants, so this has been working, but if something unforeseen happens, we do have an alternate webinar link that's available. If something happens, we'll just email that out to everyone and then resume in the webinar, but we should be fine. All right, cool. Okay, so all of that out of the way. I have the great pleasure of introducing this week's speaker. Please welcome Dr. Nina Therkildsen. Nina obtained a BA in Human Ecology from the College of the Atlantic in Maine, and a Master's of Science in Biology from the University of Copenhagen, and a PhD in Fish Population Genetics from the Technical University of Denmark. She did a postdoc with Steve Palumbi at Stanford, and currently she's an assistant professor at Cornell University, where she's a faculty fellow with the Atkinson Center for Sustainable Future and a member of the Cornell Center for Comparative and Population Genomics and the Center for Vertebrate Genomics. Did I miss anything, Nina?
I think that was a pretty comprehensive.
Okay.
Yeah, thank you very much.
Cool. So take it away, Nina.
Great. Thank you. And thank you so much all for coming. Sorry, I'm a little bit flustered. I had a huge computer failure just before this, so I didn't get a chance to make sure that the screen sharing would work. But can you guys all see my full screen now? Okay, perfect. So yeah, thank you so much all for coming. So as Brendan said, I'm going to be talking about temporal genomics perspective, in particular in relation to fisheries-induced evolution. And I guess I don't need to convince anyone who showed up to this seminar today about the power of temporal genomics, but just to kind of all get us on the same page, I'm sure what's motivating us all to consider or think about this approach is the realization that the world is changing really rapidly. Climate change and a whole host of other human impacts are drastically changing the world around us. And increasingly, that's threatening the future persistence of many species. So, of course, one obvious way to think about how we can best protect the world's biodiversity moving forward is to look at how recent changes, because a lot of these changes that we expect to accumulate over the next decades have already been ongoing for a long time. So looking to the past to see how populations have responded in the past may equip us to better protect them in the future. And genetics is, of course, really powerful in this context because DNA, as we all know, kind of works as a tag that carries on information about the past. And for this reason, just using a current snapshots of genetic variation, we can learn a whole lot about a population's history. Here's an example just showing how human demographic history has been reconstructed to quite a detailed extent, and even with some dated events. And a reason why we can, or one of the reasons why we so powerfully can infer the past is that the genetic variation accumulates these signatures over long timescales. And that can be powerful, but it can also be a limitation if we are particularly interested in impacts that have only started happening much more recently. So if we're just looking at current snapshots, we'll see the integrated effect of all kinds of different impacts that have been accumulating over thousands and thousands of years. But if we're interested in something that's only started occurring over the last few decades, it can be really hard to disentangle the effects of those things by just looking at a single point in time. So that's why by having these historical samples, we can get the real-time window into looking at exactly what is the change that's been happening from point A to point B. And of course, the more detailed we can get our time series sample, the more detailed inference we can make and reconstruct time series. And one impact that I'm particularly interested in is a question of why fish don't tend to look like this anymore. So these are cod that were caught around the 1900s in Newfoundland. And the inscription plate in the museum here says that this photo is found at, says that the fish here on the right was about 165 centimeters and weighed about 27 kilos. So for comparison, in 2018, in the survey of this, in the same area here, the largest age class that was caught in any abundance was age 12-year-old fish that were on average 85 centimeters and 7.5 kilos. So that's obviously much smaller than what used to be around. And that's in part just a demographic effect. When fishing pressure is really high, few individuals get to be very old. But it's also increasingly being recognized that there might be a genetic component. So we all know that in agriculture, breeders have been really successful at making dramatic changes to animals and plants. And the way we've done that is that breeders have every generation selected the individuals with the most favorable traits and had them be the parents of the next generations. It's much less recognized that in wild capture fisheries, we're essentially doing the opposite. We're taking out the individuals with the most favorable traits, so the fastest growing fish, the ones that are the easiest to catch, and then we let the remainder stay and reproduce to form the next generation. And when these traits that we're targeting in fishing have a heritable basis, it's conceptually easy to see. And when mortality imposed by fishing is often very strong, it's conceptually easy to see how this kind of selection pressure could cause evolutionary change. And over the past few decades, a huge body of work has suggested that fishing actually has driven very significant evolutionary shifts in a number of different traits in a lot of the world's commercial fish stocks. So here we're looking at some historical data for cod population in the southern Gulf of St. Lawrence, where on the left-hand side, we can see how the length at age 6 and on the right-hand side, an index for the timing of maturation, how both of those have really declined dramatically over the last few decades. And these kind of shifts towards slower growth rates and earlier maturation are of concern because they may reduce fish stock productivity and resilience. And as I said, a whole host of studies have looked at these extensive monitoring time series of data that we have for a lot of commercial stocks. And a review some years ago kind of summed up here. So just for cod population alone, we have very strong evidence for lots of different stocks that there really appear to have been fisheries-induced changes in traits, and for, oh, you guys can't see my mouse, right? But for lots of other species too, there's pretty strong evidence to suggest fisheries-induced evolution. But of course, even though various statistical approaches have been developed with these time series of observed trait data from the wild, it's really difficult to disentangle what component of these changes that we see are just phenotypic plasticity so that things may revert back when fishing pressure changes, and which are truly genetic changes that can be much more difficult to reverse. So I'm going to tell you today about two efforts that are two ways in which I've been using temporal genomics to try to get at this question of the genetic basis. First, looking in an experimental setup, and after that, talking about looking at wild populations. So in terms of experiment, one of the studies that really sort of got the fisheries-induced evolution debate started was this paper from 2002 by David Conover and Stephen Munch. And basically, this paper is summarized pretty well in the photo here. So these fish are Atlantic silversides, a small estuarine fish. And the individuals that you see here they're all exactly the same age. They've been raised under the same conditions in the lab. The only difference is that the size difference between them here has evolved over just 4 generations of strong size selective fishing. So what Conover and Munch did was that they established 6 laboratory populations of these Atlantic silversides from wild caught fish. And at the end of each generation, they would harvest 90% of the fish in each population. In 2 of the populations, they took out the 90% largest individuals. In 2 of the populations, they took out the 90% smallest individuals. And then there were 2 control lines where the harvest was just random with respect to size. Then they let the remaining individuals reproduce and form the next generation, keeping each population at about 1,000 individuals. And you already saw that the response to this selection was quite striking. So here we're looking at how the weight of the total harvest and the mean weight of individual fish changed over the course of the experiment in each of the 3 treatments. And you can see here that already after the 4 generations, fish from the small fish here from the populations where the larger individuals had been removed, as we typically do in most wild capture fisheries, were only about half the size or half the length of fish from the individuals where the smaller individuals had been taken out. So it was a really striking phenotypic effect. And since these were raised under common conditions, we knew that this was a genetic shift, but what Conover and Munch didn't have the opportunity to look at the time they did this experiment, just around the turn of the century. They just didn't have the tools to be able to look at genomic changes in a complete non-model species like the silverside. So that remained a mystery. But fortunately, they had the foresight to just take all the fish from the experiment and throw them in bags and keep them in the freezer, so that we were able to, about 5-6 years ago, we were able to go back and get these fish and extract them and use modern sequencing technology to start looking at the genomic underpinnings for these dramatic shifts observed in the experiment. And as I said, the silverside is a complete non-model species. We're really starting from scratch here. No prior genomics work had been done, but we were interested in using an approach that would let us scan the entire genome so we could get the most comprehensive view of the genomic shift. So we used a low coverage whole genome sequencing approach, which I'm very excited about. So if anyone wants to talk about that anymore, please don't hesitate to reach out. Because what we're finding is that it's only slightly more expensive than, for example, PoolSeq. At the time it was about $50 per sample. Now we can do it about $20 or $30 per sample. But we get, by individually barcoding each library, we get much more versatile data. And also, just as a little plug here, we just recently put together a guide, sort of a beginner's guide for people who are interested in considering this low coverage whole genome sequencing approach as an alternative to, say, RADseq or PoolSeq. So check that out if you're interested. It's up as a preprint. But what we did here, when we started this work, we didn't have a reference genome for the silver side. So we mapped the whole genome reads to a reference transcriptome. So we were kind of doing an exome capture in silico. And we sequenced a total of 850 individuals, each to about 1.3x coverage. And based on that, generated data for about 2.35 million SNPs. And because our coverage is so low, all the analysis that I'll be talking about here is done in a probabilistic framework that's based on genotype likelihood. So we don't call genotypes, but we take the uncertainty about the actual genotype calls into account in all the downstream analysis. And all this work on the silver-side experiment here was done in very close collaboration with a former postdoc in my lab, Aaron Wilder, who's now a conservation geneticist at the San Diego Zoo. So just to give an overview of our sampling, so the experiment was established with about 700 wild-caught individuals. So we took 75 individuals out of that source pool used to establish the experiment, generation 0, and then we sampled about 50 individuals from each of the 6 populations after the 4 generations of size selection. Sorry, just hold on 1 second, I think that's some noise. Okay. And once we started looking at the sequencing data, the first thing we noticed was, as expected, that there was a really big drop in genetic diversity over the course of the experiment. So here is a measure of genetic diversity on the y-axis, and the gray bar is generation 0, and the colored bars are the different lines. So as expected, when we take a limited number of individuals and breed them over several generations, there's going to be a lot of drift. So we lost a lot of diversity. But what was interesting was that both the up select, so all of the size selected lines, so both the up and the down selected lines, lost more diversity than the 2 control lines. So that is suggesting that the size selection accelerated loss of genetic diversity. And part of that we saw from just looking at the number of unique mitochondrial haplotypes and estimates of relatedness. We saw part of that effect was that it lowered the effective population size in those populations more than in the control lines. But we're also of course expecting that selection would have played a role in accelerating those changes. So then the next challenge became then, how do we disentangle what of these changes are just drift and what might be selection? So a fair expectation seems that selection would favor similar alleles in the similar treatments. So we computed a statistic that's the minimum allele frequency difference in the same direction between up and down selected replicates. You can basically just think of it as how much parallel allele frequency change was there. And we did a number of simulations of populations that showed the same amount of change as we saw in the experiment. And consistently we found that when we compared this diffstat statistic to our observed, we consistently saw quite strong enrichment of parallel change in our observed data. So for example, if we discounted the number of SNPs that had a diffstat value greater than 0.3, so basically showed a greater than 0.3 allele frequency shift in the same direction in both replicates, We never saw more than 6,000 SNPs doing that in any of our random simulations, but we saw more than 10,000 SNPs doing that in our observed data. So there was clear an enrichment. Some of this is still going to be just by chance, but some portion of it is also likely driven by selection that's consistent between the replicates. And to further support that, when we computed a kind of a polygenic score, we summed an allele score across the top 1%, the SNPs in the top 1% of this SNP statistic, we saw that this allele score was significantly associated with individual fish length within each of our treatments. So by design, we expect it to be correlated between treatments, but the fact that we also saw within each of the group a correlation suggests that there actually is an effect of these combined. So a polygenic effect, tiny effect from each individual SNP, but across these thousands of SNP, there is some effect on the size. To look further into interpreting these results, we also started looking to wild populations of silversides. Because silversides are distributed all along the east coast of North America. And this is actually one of the deepest thermal gradients in the world, the coastal waters here. And we might expect that an ectotherm like the silverside would then also show very different growth rates, that they would grow much more slowly in the north where the water is cold, so they wouldn't get as big, and would grow much better in the south. But when you go out and sample silversides from the wild, they reach about the same adult size all up and down the coast. It's a very different picture though when we take them into the lab. So this photo here, we kind of try to emulate the photo from the experiment, but the fish that you see in the photo here on the right with the light background, those are individuals of exactly the same age that we've raised under common conditions in the lab. The only difference being that the larger fish and the top have parents coming from New York, and the bottom fish have parents coming from Georgia. So what we can see here is that there's this really strong genetically based difference in growth rate that only becomes apparent when we take them into the lab. And extensive work by David Conover, who was a professor at Stony Brook University and spent much of his career studying these silversides, has demonstrated that the capacity for growth that only gets revealed once you take them into the lab is very tightly associated with latitude in these silversides. And extensive field and lab work that he's done has shown that the selection pressure is that because of the short growing season in the north, there's very strong selection for the fish to reach a certain size before winter. And then there's that trade-offs with predator avoidance, causing slower growth in the south. But this kind of selection pressure for the faster growth creates this interesting pattern of variation called counter-gradient variation, where basically the genetic influence on the trait oppose the environmental influence. We would expect fish to grow slower in the north, but then they have evolved genetic compensation to be able to still grow faster. And this is a really interesting type of variation because it actually results in reduced phenotypic variation. So it's kind of a cryptic adaptive divergence, which I think also has some bearing when we start talking about the fisheries-induced selection signatures. But in that case, so we looked to these wild populations of silversides. And initially in our initial study, we had included samples from 4 different locations along the cline, as you see here in the top left corner. And when we sort of took the intersection of SNPs that showed very strong geographic differentiation, and also were in the top of those SNPs that showed parallel changes in the experiment. So when we look at an intersection here of 357 SNPs, and then the goal was here to just look at whether there's any commonality between what varies between fast and slow growing silversides in the wild and fast and slow growing silversides in the experiment. So this subset of SNPs here, what we're looking at the y-axis is the change in frequency of the northern faster growing allele. So we see that for the two green control lines, there was basically no particular directionality, but we saw that for the blue, the up-selected lines that were selected for faster growth, there was a very strong tendency for those to have, but it was the northern allele that had increased in frequency. And for the yellow and orange, the down-selected lines, it was for the vast majority of this SNP set, the southern slow-growing, naturally slow-growing allele that had increased in frequency. So this way we see that in the experiment actually recapitulated these patterns of variation in the wild. And mind you that this is across these pretty large hundreds of SNPs that are distributed over the entire genome and show very little linkage disequilibrium. So it's suggesting a very highly polygenic response in which the experiment kind of recapitulated what was going on in the wild. So we could kind of think of the selection going north and south, fish north and south. But what was really interesting was that this parallel signature was actually not the most predominant. So when we, in this plot here on the right, we plotted the FST, so the amount of genetic change from generation 0 to generation 5 in replicate 1 on the x-axis and in replicate 2 on the y-axis, we see that if there was a lot of parallel change, we would expect high differentiation SNPs to kind of align along the diagonal. But what we instead see is that, especially for the down-selected lines here, we see kind of a lot of SNPs piling up against the axis, suggesting that these SNPs changed a lot in one of the replicates, but not the other. So it actually looks like the predominant pattern here was more one of idiosyncratic allele frequency shifts rather than shared shifts between the replicate subjects to the same treatment. And that also becomes clear if we start looking at how these SNPs were distributed in the genome. So we didn't have a good genome to start with, but we anchored our transcriptome onto the chromosome-level assembly of a related species, the Medaka, to get a sense for genomic location. And then here on the y-axis, we have each SNP, for each SNP, we have a measure of how much allele frequencies changed between generation 0 and 5, and then each of the selection lines. And we can see here in the two control lines, there wasn't anything too notable going on, just pretty low levels of allele frequency shifts across the entire genome. In the two up-selected lines, we see a bit more allele frequency change, and we also see some tendency for clustering in particular regions. But interestingly here, we are already seeing that clustering that happens in, for example, the up one line is not necessarily evident in the other up-selected line in the same location. This was most prominent in this chromosome 24 location here, where we saw a major change in one of the down-selected lines, but hardly anything going on in the other down-selected line. So what was going on here was that there was a set of more than 9,000 SNPs that were in super strong linkage disequilibrium. Basically, they were acting like a completely linked block that in generation 0, we just had a single copy of an alternate haplotype, and that haplotype came to a frequency of about 0.6 in generation 5. So just a massive change in frequency over these few generations. And if we look back to the wild silverside populations, these 9,000 SNPs actually also show completely fixed differences between the three northern populations. So these histograms here are showing the allele frequencies, sort of the count of SNPs at different allele frequencies. So almost all of these SNPs were fixed for one allele in the three northern populations and fixed for the opposite allele in the southern population. And given that the genome-wide level of differentiation is quite low, which I'll get back to later, these are likely under very strong divergent selection in the wild as well, among these naturally different growth rate individuals in the wild. And we also saw that this haplotype explained a lot of the variation in individual lengths, so further supporting its role in this response to the size selection. So if this haplotype was so important for the response to selection in one of the down-selected populations, why did it not do anything in the other one? What we think happened is that since the experiment was established from New York, so this middle location here, where the southern naturally slow-growing haplotype is really rare, we think that probably just by chance, it didn't get sampled, it only got sampled into one of our replicate lines and not the other. So selection didn't have the option to act on it in that other line. But then what's super interesting is that if we're looking at the phenotypic shifts, we actually saw very similar responses. So what's shown in the error bars in this plot here is showing the range of the means between the two treatments. So the phenotypic response was pretty similar in these two populations, even though the genomic shifts were completely different. So this is really suggesting that there were multiple genomic pathways to the same phenotypic outcome. And one of these down-selected replicates, a big selective sweep seemed to have played a major role, where the other populations was able to respond just as strongly without that. So conclusions that we draw from these initial results from the experiment is that in this experiment, it seemed like the size-selective fishing accelerated loss of genetic diversity overall. We also saw the highly polygenic parallel allele frequency shifts that recapitulated responses to natural selection. But then on top of that, there were those idiosyncratic genomic responses that were underpinning what seemed to be a parallel response at the phenotypic level. So kind of our take home from this was that parallelism at the phenotypic level may mask highly divergent genomic shifts. So just because it looks like human impacts of fishing in this case is having the same effect on different populations, it might have really different population genomic consequences. And also for that reason, impacts can be unpredictable. So where do we take this next? We still think, so looking just at the first peek we got at these cell-site populations, we're just looking at 4 locations. We were very intrigued because since we know that they vary genetically in the trait that was under selection in the experiment, and then we're seeing when we're looking, so this is now looking genome-wide at the patterns of differentiation between different pairwise persons. So we're seeing that it's not just on the chromosome 24 that we have this block of fixed differences between different locations, but there are at least 3 other places in the genome, plus a lot of other stuff going on. But this was, of course, all a little bit messy because we didn't have a species reference genome. So we were, to some extent, like our conclusions here are limited or based on the assumption that synteny is conserved between the medaka that we had anchored everything to. So we've now gone ahead and gotten ourselves a species chromosome, Atlantic cell site chromosome-level assembly. This has been led by another former postdoc in my lab, Anna Tigano. And we saw, fortunately, that the synteny is relatively well conserved with the medaka. And also another interesting thing we saw was that actually, as we did some deeper sequencing of a few individuals, we saw that silversides actually has the highest level of heterozygosity observed in any, or reported for any vertebrate genome to date, which may also perhaps explain part of why it shows such a remarkable adaptability. But in addition to that, another former postdoc, Arne Jacobs, then went back to our original data and added data from several other locations and map that to our new genome. And we see here, so these are comparing all of these other locations to the southern Georgia locations. And we see that actually mapping to the species genome just makes the patterns much tighter. That we still see these big blocks popping out, and we see kind of various levels, very low levels of background differentiation among many of these populations. A grad student in my lab, Maria Kopian, and our collaborator Hannes Baumann at University of Connecticut have also done some comparative linkage mapping to look at what is really underpinning these big blocks of differentiation. So she's set up crosses between fish, so both some pure crosses between fish from Georgia and New York. So we could both look at pure maps and hybrid maps. And when she mapped those genetic maps to our reference genome, which is from a Georgia. So a Georgia linkage map mapped to our reference genome, which is based on a Georgia individual, we see a very collinear relationship for this chromosome 24, as we might expect. But when we map the New York map, we see very clearly that this block of high differentiation is really a big inversion. And when we look in the hybrid, we also see that, as expected, recombination is completely suppressed in this inverted region. And the same is true for all these other, for these, at least these two other major blocks of differentiation. They're also underpinned by inversions, and we have additional evidence from high-C data and alignment of genomes to really confidently know now that these truly are inversions that coincide with the differentiation. So Maria has also mapped a number of different traits, including growth rates. And as we already were expecting, at least for chromosome 24, she's finding that to be a strong QTL for growth rate, but also she identified 5 other QTL, 2 of which coincide with other inversions that we know exist. So these QTL explain about 7 to 12% of the variance in growth rate, which is perhaps quite high for something we might expect to be really polygenic like growth rate. And also the effect is so strong, and we already saw that from the experiment, but when we look at our samples from the wild, like out in the wild where all kinds of environmental variation is going on, we can still see a noticeable difference that individuals that carry, that are northern homozygous for this chromosome 24 inversions have notably greater length than other individuals within the same populations. So this is kind of tying this back now to the experiment. This is bringing back to, so now we know that there are these big inversions that have a very strong influence on growth rate in the wild, in combination with all the polygenic signatures that we've also identified. So then if we just think back, this experiment here was established with fish from New York, where we can see that we actually have very little variation. So here you can kind of see the northern and the southern haplotypes in each of the populations sampled here. So where the experiment was established from, we see almost exclusively either the northern or the southern haplotype. So kind of an interesting thought experiment is how different would the outcome have been if the experiment had been established from this location a bit further south, where we, for 2 of these major growth rate QTL inversions, see both the northern slope fast growing and the southern slope growing haplotype being pretty abundant from the beginning. Would we then have expected consistent sweeps in all replicates, or could there still be a case where the signature was more polygenic? And then what if it was established from up here in the very north, where we don't see any variation at all in these haplotypes, growth rate inversions? Could we imagine that the genomic architecture underlying growth variation up there is very different, and what kind of response would we see? So that's definitely a very tempting experiment to be setting up, but also, of course, be a big logistics ordeal. So for now, just a thought experiment. But in any case, it seems that the responses to selection really could depend very much on standard genetic variation. So this is what goes on in the lab. What about looking to real fisheries? So we actually have a pretty unique opportunity to use a temporal genomic approach to look directly for genomic changes in commercial fish populations, because for a lot of commercial stocks, there are these otoliths that you see here. These are little ear stones from the inner ear fish that, just like trees, form annual rings. So they've been collected by fisheries biologists along with scales that also can be used for aging. to kind of estimate the age composition of fish stocks. So they've been collected since the beginning of the 1900s. And in some cases, these collections have been kept. So this is a photo here from the Greenland Institute of Natural Resources, where I've done a lot of work earlier, where we can see that the collections go all the way back to the 30s. So these otoliths don't actually contain much DNA themselves, but the little bit of dried tissue, mucus and blood on the surface has enough DNA that we can work with. So this means that these kind of collections of archived samples are really just a treasure trove of information about the past that's just waiting to get uncovered. There have been a few studies looking at changes across individual markers, but it's really only now that we have the technology to be able to take advantage of these collections to look at what's been happening genome-wide in these populations. So the first whole genome study of temporal change in fish populations just came out a few months ago. Some of you may have seen it from many esteemed colleagues here, led by Emil and Pinsky, where they were concluding that in the samples they looked at, they did not see any evidence of major genomic sweeps or selective sweeps, even though they were looking at. So basically what they did, they had endpoint samples, so a very early sample from 1907 in Norway and 1940 in Canada, and then comparing to a more contemporary sample. And both of these populations have been really heavily overexploited, and monitoring data suggests that they both suffered a strong shift in the age maturity. But what the Pinsky team found here was that here showing patterns of differentiation over time in Canada and Norway, and really not much kind of popping out, no big sweep signatures. There were a few potentially interesting shifts here and there, but not the kind of major shift that we saw in one of the silverside populations, for example. So what does this mean? In some ways, it could be tempting to conclude here that there hasn't been fisheries-induced evolution going on here, and that those phenotypic shifts are just caused by density dependence or various other plastic effects. But remember, if we go back to look at what happened in the silverside experiment, we actually saw that one of our down-selected lines ended up showing just as strong a response to fishing as the other one, without showing any major selective sweeps. So it clearly seems possible, just from the experimental evidence here, for there to have been quite substantial fisheries-induced evolution going on without it necessarily leaving signatures that can be picked up in a scan with relatively small sample sizes. And also, these findings from the Pinsky study contrast somewhat with work that I did as part of my PhD now quite a few years ago, where I was looking at some temporal samples from this southern Gulf of St. Lawrence cod population that I showed you these shifts in the temporal change earlier on. So I was able to get samples going back to 1928 from this sample, and for this location up to 2008, with approximately 30 individuals from each year. And we also were able to get samples from 3 other surrounding populations, just to be able to get a little bit of spatial context for what was going on. And just so this is just a little bit further south than the cod population studied in the Pinsky paper. And this is, of course, a much more limited approach. It seemed extremely cutting edge at the time that we could look at more than 1,000 SNPs, but now, of course, it's almost archaic that with this small marker set. But in any case, we used a genotyping assay to genotype up to 1,000 SNPs. And then our goal was to look at temporal change and identify SNPs that showed greater allele frequency change over time than expected from drift and sampling error alone. And when we first compared just the earliest and at that time current point from this 40, the Gulf of St. Lawrence population, we saw that most of the SNPs didn't show much change over time, but there were some that showed much greater change. So along the y-axis here is a measure of genetic change over time, much more change over time than we would have expected just from drift or sampling error. So then we were, of course, curious to understand what's been happening in this intervening period. But when we then went ahead and genotyped in these intermediate years, we saw that for this set of SNPs that seemed to have showed big change, all the change seemed to have happened before 1960, and then things have remained pretty stable since then. And the same was true in all the surrounding populations. When we then looked at just from that period from 1960s onwards in each population, we found a different set of temporal outliers in some of the populations, some that showed greater allele frequency change than we would expect. But again, it was different set of SNPs in different populations. So don't worry about any of the names here, but the point here just being that it's different SNPs that are highlighted in blue as temporal outliers in the different populations. And because things change differently within different populations, of course, the spatial patterns were, so because we had somewhat similar year or time period sampled in different populations, we could also do spatial outlier scans here in 3 different decades. And when we look at what were the loci that popped out here as being the most highly differentiated, of course, many of these are probably going to be false positives, but at least if we just think about what are the most highly differentiated, then we also see that there was no single SNP that showed elevated spatial differentiation throughout the entire period. There was kind of a shift in which set of SNPs were spatial outliers at which period in time. And it kind of makes sense that actually the SNPs that were temporal outliers were also the ones that were spatial outliers at some point, but then things changed over time in some populations and not others. So we end up with this really complicated picture here of really strong spatial temporal interactions and very dynamic system that's a little bit hard to make sense of. But one thing to note is that a lot of the SNPs that were outliers clustered in 2 blocks on 2 linkage groups of chromosomes that we now know, we didn't know at the time, but now we know that those are also inversions. So this actually has a very similar genome structure to silversides with 4 large inversions on different chromosomes, and 2 of these are variable in Canada. And we know from lots of studies that have looked at genomic variation of cod populations across space, that these 4 inversions show highly elevated level of spatial differentiation. So the 2 that the temporal outliers here, one of them is this linkage group 12 that has been shown in prior studies to have allele frequencies correlate pretty strongly with local sea surface temperatures. And if we look at the patterns of change within our focal population, we saw kind of a fluctuating up and down signal here. And that actually happens to correlate somewhat with a local temperature experienced by cod. And similarly, the other one on linkage group 1 has previously been linked to life history variation, and that showed kind of a clinal change here that in some ways looks pretty similar to the shift that's been happening in maturation index. So it's possible that some of these shifts actually have to do with either temperature or fisheries-induced selection. But of course, with such few time points as we have here and the limited number of markers, it's hard to draw really robust conclusions. But a grad student in my lab, Nicholas Liu, is now following up and doing whole genome sequencing on some other samples from the same population. So he has samples from the 1960 to 2018, and we're just starting to make sense of the data. So we only have preliminary results right now, and we need more sequencing depth before we can say anything with certainty. But just from kind of an initial first glance of change over the whole time period, looks in some ways a bit more like what Pinsky and his team saw in terms of somewhat limited change going on, and especially for some certain time periods, it's even less. But between other in time intervals, we actually see some elevated differentiation that could look like more interesting patterns. And in particular here, we see again that temperature-associated inversions showing up. And interestingly, also this linkage group 18, a region that we have in another study of cod in the Gulf of St. Lawrence, where there are separate winter and spring spawning groups that use the same spawning grounds. We found that same peak on chromosome 18 to be very tightly associated with spring versus winter spawning. So the fact that that is showing up in some of our data as temporal change, and interestingly, looking back at Malin's paper here, it actually shows up, that exact region actually shows up as a bit of an outlier in their Canadian data too. So there could potentially, even though we don't see these huge striking sweeps, there could potentially be some temporal shifts that would be interesting to follow up on. So I'll wrap up here and just sort of with my take on this is that the jury is still out on whether fishing or recent climate change has caused evolutionary change in wild fish populations. I think there's a lot we can learn from now being able to do whole genome sequencing on several different populations and more fine scale time series. It might not be that it's all a linear change, but there could be some interesting things in the fluctuations. And then I also think what we're learning from the Silverside work is really that the genomic architecture and the local standing variation very likely will influence the kind of genomic patterns that we'll see in response to selection. So we could see a very different genomic signature in based on the same, or in response to the same selection pressure on different populations of the same species, for example. So with that, I'd just like to thank all my collaborators. I'm so grateful for the amazing team I get to work with, and then I would be happy to take any questions if there's time.
Okay, thank you, Nina. That was great. Let's see if we have we don't have any questions in the chat yet. Let's see. Yeah, I think I mean, one thing that I was thinking about was just those idiosyncratic allele frequency shifts, especially in the experimental evolution study. Thinking, well, maybe we would see more parallelism in a large wild population with lots of migration. Do you think that any of those? Would you expect to see something like that, or how do the COD studies kind of inform that? Like, is that idiosyncrasy in those experimental studies where you have smaller population size? Is that because of the smaller population size, or would you expect to see those idiosyncrasies in big wild populations too?
Yeah, I think the couple of things. So I guess at least what we're seeing in the Silverside is that it seems to be that something like growth is just so genetically flexible that there's so many different genetic variants that can affect growth. So I guess in large populations that would have the same kind of standing variation I mean, yeah, I guess it's hard to say. When you have large effect variants, you'd expect selection to be stronger on those and those to change more. So I guess I would assume whenever those were present, those would probably play a strong role. But I think a huge complication with looking at the temporal shift is also this interaction between temporal and spatial change that I think is always going to be the case, because a lot of these variants that get to be under selection seem to have some functional relevance, right? So then they're also likely to already be under different selection pressures, different locations. So then when you sample a population, when you sample a species in the same location over time, are you then sampling sort of direct descendants of the same population? So you're looking at evolutionary change within a population, or are you sampling populations moving in and out of the same area, so that you're looking more at a spatial shift in what populations inhabit what area. I think that's a thing that gets tricky to sort out when, as is the case for a lot of marine species, that genome-wide there's almost no genetic differentiation, and really the only differentiation you see are in these functionally important regions. So
cool. And my other question that I was thinking about was, does this say anything clear to you about how we should fish, and like what are are there fishing practices that would be like, you know, now that we know that about now that we know how they're potentially causing these, you know, selective, you know, shifts in these species, does that can you make any clear conclusions about how we should change those practices?
I think, I mean, in some sense, the good news is that what I think helps counteract fisheries-induced evolution, or what is good in terms of fisheries-induced evolution, is also what's good in terms of what else we know about how to manage fisheries well. So reducing fishing pressure, making sure that individuals get a chance to spawn, and so on. And so I think it's overall, I think the point that they're making in the Pinsky et al. Paper, one of the key points is that in some ways, even if there might be some fisheries-induced evolution going on, there are bigger problems in terms of just outright over exploitation, that if we take care of those, we'll also automatically take care of minimizing fisheries-induced evolution. But I think in terms of, the big concern about fisheries-induced evolution is the reversibility. So if it turns out to be primarily a polygenic response, so that in that case, you just see small allele frequency changes across lots of loci, you don't necessarily erode any key critical variants that you need to reverse the trait, then that's really great news in terms of not causing irreversible harm. But if a lot of other species have a similar architecture, underlying growth that silversides seem to have, where these large inversions seem to play a really major role in controlling growth, I think there's much more potential for potentially eroding important genetic variation.
Okay, great. And we have a question from Andrew now. Andrew, you can go ahead and ask that.
Yeah, hi. Thank you so much for a great talk, Nina. That was just super. Obviously, the data are great. The presentation's outstanding as well. I really like the point that you made that even though phenotypes can appear very similar across, you know, this gradient along the Atlantic, that there's a lot going on under the hood there in terms of, you know, strong diversifying selection to keep things the same. You know, that's really made a strong impression. And so I'm also really, you know, we read the Pinsky paper as a lab group. It came out like literally a week before I teach like this stuff about sort of fishery-induced evolution, one of my classes, and the COG was like classic example with the life history, you know, changes over time, and I had to quickly like rethink like how I was teaching that at the last minute. But because that was a big surprise that they really found no really strong signatures of selection. And, you know, I kind of like the explanation that you're sort of gravitating towards, that's like perhaps highly polygenic. But can somebody do the common garden experiment, like to test whether it's plasticity or not? Obviously, you can't resurrect like 1940s fish, but is there a variation in contemporary within between contemporary populations where people could say, you know, we could figure out at least at the phenotypic level, is this are these heritable differences? Is this heritable variation that we're seeing here?
Yeah, I mean, I think it really, for us to see these kind of big selective sweeps, that have to be some major effect loci, right? Otherwise, we wouldn't see such huge allele frequency changes. And I think in cod, even though there's been a lot of studies documenting the elevated levels of differentiation at these inversions. I think it's, of course, cod are just much harder to keep in the lab than silversides, and to raise for multiple generations to be able to really get at a sound understanding of the genomic basis of maturation and growth. So I don't think we know yet what to expect with cod. So I guess I don't know if what we're seeing in silversides is really unusual, or if there actually are a lot of fish out there that would have that similar type of genomic architecture for traits that might be targeted, or if silversides are just weird. And in most cases, I mean, sort of with classic quantitative genetic theory, we would expect something like growth and maturation to just be highly polygenic and maybe have a few larger effect, but mostly lots of small effect. So yeah, I guess I don't have a very good answer. And I think it'd also be interesting to, a lot of the fisheries-induced evolution work is so centered on cod that it would also be super interesting to look at other, but it's clear, like if we don't see it in cod, and they looked at some of the most sort of textbook examples of where we think fisheries-induced evolution should have happened. So if they don't see the changes in those particular populations they looked at, then unless it's because cod has a very different genomic architecture than other species, I don't know how widespread it will be.
Cool, thank you.
Okay, so unless we have any other questions, I think all that we have left to do is give Nina another round of applause for a great talk. So yeah, thanks, Nina. And yes, that was great. And just a quick note for people who are still here. Our next seminar, we usually have these on Thursdays, but our next seminar is a Friday seminar on July 18th, I believe. I encourage everyone to come back for that. And yeah, great. Thank you everyone for coming, and thanks to Nina. And I hope everyone is having a good summer, and have a good day. Thanks.
Thank you very much.